Thank you so much for that introduction, and thank you for the opportunity to share some thoughts with you. Today, I will give a talk on bioelectricity with a specific focus, although we don't generally focus on microbiology or immunology in our group. We've done a little bit of work in those areas, and so I think this may be of special interest to this audience. Here are the main points that I'm going to transmit today. I'm going to talk about the fact that numerous issues in biomedicine depend on two critical things. One is the ability to communicate anatomical and physiological target states, aka goals, to cellular collectives, to groups of cells. And also the correct setting of the boundary between self and world. And this is very important, and I'll show you what I mean by this. I'm going to tell you that biology uses natural bioelectrical networks to establish emergent agents that have pattern memories, they have problem-solving competencies, and they maintain a border between themselves and the outside environment. And I'm going to argue that cell groups are literally a collective intelligence that navigates some very interesting spaces, including the space of gene expression, the space of physiological states, and of course, the space of anatomical structures. And failure modes of that process result in things like birth defects, they limit regeneration after injury, they result in cancer, possibly immune disorders, possibly aging. This is a cutting edge work, we don't really know that yet. But we're going to be able to find out soon because tools are now available to read and write the content of the protocognitive medium of which we are all made, these bioelectrical networks among cells. And that offers a roadmap for a new kind of regenerative medicine approach that targets the software of life and complements all of the work in today's molecular medicine on the hardware of cells. So the talk is going to come in three parts. An introduction where I set up some of the unusual perspectives that we use on all of this, and then we'll talk about bioelectricity as a kind of cognitive glue that keeps all of these cells working together. And then at the end, we will talk about this notion of hacking as a fundamental concept in biology. So let's start with the simple observation that all of us were a single cell once. So each of us made this amazing journey from something that people call just chemistry and physics. And I really don't like that term at all, but when you look at a single unfertilized oocyte, what you see is a little chemical system, and you say, okay, that's generally well described by chemistry and physics. And then eventually we become something like this, which is a system that has its own inner perspective. It has all kinds of intelligent competencies. It has amenability to techniques of behavioral science and maybe psychoanalysis and things like that. And so this remarkable process of embryonic development takes us from physics and chemistry into the science of the mind. And we all made that journey slowly, step by step. There is no magical lightning flash at some point within that process that converts a purely physical system into a truly cognitive system. Okay, there's no special point at which that happens. This is a slow and gradual journey. And all of us are kind of collective intelligence because we're all made of things like this. This is a single cell. This happens to be a free living organism. This is called the lacrimaria. It has no brain, it has no nervous system, but you can see that it's quite competent at all kinds of tiny little cell level agendas. And so maintaining its metabolic goals, its physiological goals, its anatomical goals, and so on, it's quite good at it. And the interesting thing about biology is that each of us is made of this multi-scale competency architecture that is not just structurally, we're sort of nested dolls where groups of us are made of organisms, which are made of organs and tissues and cells and subcellular components. But each of these layers is actually solving problems in its own space. These are not merely passive subcomponents. They are all competent in solving various kinds of problems. Now, as humans, we're reasonably good, not great actually, but reasonably good at recognizing intelligent behavior of medium-sized objects moving at medium speeds in three-dimensional space. So primates, maybe birds, maybe an octopus, maybe a whale, we have some ability to understand and detect intelligence when we see it. But there are all these other problem spaces in which various agents live and navigate. There's the space of possible gene expressions, very high dimensional space. There's the space of different configurations of shape of the body, and then of course physiological space. We're going to spend most of our time talking about anatomical morphospace. But just imagine if we had evolved with a built-in sense of our own blood chemistry. So if you had some sort of a sensor, like almost like a tongue, but let's say 20 different metrics that you could take of your blood chemistry at any given time, and you were able to feel you had a sense organ that was able to report that. I think we would have no trouble perceiving that we live in a 23-dimensional space, and that our liver and our kidneys and our various organs are actually intelligent agents navigating that space to solve all sorts of new stressors and problems that come at us every day. And so this idea that we have to get better at navigating these different spaces and understanding how other systems navigate these spaces, and recognizing exactly what competencies do they have to make decisions to move through these other spaces, I think is going to be a critical part of the medicine to come. And this is long before you get to brains or anything like that, we find out that even subcellular components, right, that even individual cells are already a collective intelligence themselves, because they have molecular pathways and gene regulatory networks. And as you can see here, these molecular networks are themselves a kind of learning agent. They have 6 different kinds of memory that they can muster, including Pavlovian conditioning. And that's before you even get to the level of a single cell. So it's sort of this multi-scale architecture of agents with really interesting properties that we want to take advantage of. And for our focus on navigation of anatomical morphospace, let's ask this question. Here's a cross-section through a normal human body. You can see the amazing complexity and order here. Everything is in the right place, right next to the right thing, the right size, and so on. And one might ask the question, where does this pattern come from? In other words, where is it encoded? It's a very specific pattern. It's the same in most individuals. Where is it encoded? And we know that we have to get there from this kind of starting position. So we're all we all start as a ball of embryonic blastomeres, and eventually you get something like this. So where is it stored? Well, initially people will say, well, it's in the DNA. Of course, it's in the genome. We know where it is. It's in the human genome. But we can read genomes now, and we know what's in the genome. Nothing like this is directly encoded in the genome. What the genome does specify is protein sequences, which are the tiny hardware that every cell gets to have. But beyond that, there's this process of physiology where, using that hardware, these cells have to know what to build. They have to know when to stop. As workers in regenerative medicine, if a part of this is missing, we would like to know how to convince the cells to regrow it. And something I won't have time to talk about today, but as engineers, we ask the further question, which is, and what else can we get these exact same cells to build? Same DNA, same cells. What else can we make them build other than this? What else are they willing to build? So these are some of the big questions that we're asking. And this comes up a lot. The structure of the termite nest and the structure of the spider web are not directly in their genomes either. And so all of this is a working out of a kind of physiological process that is running on top of the hardware that's genetically encoded. And so as we think about the future of the field, the end game, here's how we think about it. Someday, you will be able to sit down in front of a device, a system called an anatomical compiler. And the idea is that you will be able to draw, like this, any animal, plant, organ, biological robot, anything you like. You'll be able to draw it. And if we knew what we were doing, the system would compile down this anatomical goals description into a set of stimuli that would have to be given to cells to build exactly what you want it to build. In this case, this nice 3-headed flatworm. So understand the idea. This is complete control over growth and form. You should be able to say what you want and know what are the signals that you have to give to cells to produce it. Now, this is not only fascinating from the perspective of evolution and cell biology, but it's critically important for biomedicine, because if we had something like this, all of these things would go away. So birth defects, traumatic injuries, cancer, aging, maybe degenerative disease, all of this could be handled if we could communicate to a group of cells what it is that they should be building. And so the key is that this anatomical compiler is not a 3D printer. It's not about putting cells where you want them and micromanaging their properties. It's really a communications device. It's a translator of your anatomical goals to the set points that these cells are going to try to build. And now I'll point out, we don't have anything remotely like this. We're very far from this. And you might wonder why. I mean, genetics and molecular biology and genomics and so on have been operating for decades. Why don't we have something like this? I just want to show you a very, very simple example of what I think is missing here. So this is the axolotl larva. It's a kind of aquatic amphibian, and the baby axolotls have little forelegs. This is a tadpole of a frog, let's say Xenopus laevis. They do not have legs at this stage. In my group, we make something known as a frogolotl. A frogolotl is a chimeric embryo. It has some tissues from here and some tissues from there, and they're perfectly viable. And now I ask a simple question. We have the frog genome. We have the axolotl genome. So we've sequenced, both of these animals have been sequenced. Could you tell me whether frogolotls are going to have legs or not? And the answer is no. At this point, we have absolutely no models that will tell us, allow us to predict from the genetic data whether this frogolotl is going to have legs or not. And that's because while we are very good at, as a community, at getting this kind of information, so hardware, really nano level hardware information about what genes talk to what other genes, what proteins associate with each other, and so on. While we're very good at this kind of information, we're really still quite a long way from having any kind of control over the large scale decision making that cells have in order to reach anatomical set points in that morphospace. You know, how do individual cells agree on what they're going to build? And so I think that something that's very instructive is the journey that information technology took. So this is what computer programming looked like in the 40s and 50s. You literally had to interact with the hardware. That was the way you were going to program this machine. But the reason that nowadays, when you want to, on your laptop, switch from Photoshop to PowerPoint, you don't get out your soldering iron and start rewiring, it's because what computer science realizes is the power of a reprogrammable medium. The idea that if your software is good enough, if your hardware is good enough, and I'm going to argue that biological hardware is definitely good enough, then you have access to some really interesting competencies of your machine that don't require you to be doing everything at the hardware level. And modern molecular medicine is basically pretty much focused on this level now. So CRISPR, genomic editing, pathway rewiring, protein engineering, all of these things, all the excitement there is all about the hardware. It's all about the molecules. And I think we're really leaving on the table something very important, which is the ability of different levels of biology to process information in a way that we can correctly call intelligence. Now, what do I mean by intelligence? I'm using William James's definition. There are many, of course, definitions. I like this one. What James said was that intelligence is the ability to reach the same goal by different means. So it's a nice cybernetic definition. It doesn't talk about having a brain or what scale you are temporally or spatially, or what space you're operating in. It's this idea that as an agent, you have goals and you have some degree of competency to get those goals met despite various things that are going to happen. And he talks about a spectrum of systems from two magnets trying to get together. And this is incredibly kind of low down on that spectrum of competencies, because, for example, if you separate them by a piece of wood, the magnets will never go around and join each other, because that would require them to temporarily get further away from their goal, and they simply don't have the capacity for that kind of delayed gratification. Romeo and Juliet, on the other hand, do. They have planning, and they have all kinds of other things they can bring to bear on their problem. And in between, you have autonomous vehicles and various kinds of animals and cells and tissues. And so this is the kind of a spectrum of intelligence and various competencies to solve problems in a kind of space. So let's ask, what kind of collective intelligence do cellular swarms deploy? So cells in the body, what can they actually do? Well, the first thing to think about is that development, while it's very reliable, so most of the time this egg develops into exactly what it's supposed to be, it actually isn't hardwired, because you can cut early embryos into pieces, and each half, so let's say you divide it in half here, each half, you don't get a half a body, you get two perfectly normal monozygotic twins. And so you've got this ability to navigate to the right ensemble of states, this target morphology that we call a normal human, within normal human variation. You can navigate there from different starting positions, as a complete embryo, as a half embryo, and so on. And you can even go around some local minima along the way. So that's one example. That kind of thing is not just for embryos. Some, like here's that axolotl again, these guys regenerate their limbs, their eyes, their jaws, their tails, including spinal cord, portions of their brain and heart. And what you can see here is that you have this salamander limb. They do this natural experiment where they bite off each other's legs if you house them together. And so no matter where it's been amputated, it will do exactly what's needed to grow, and then it stops. That's the most amazing thing about regeneration, is that it stops. How does it know when to stop? Well, all of these cells stop their activity when a correct salamander limb has appeared. So what you're seeing here is a kind of error minimization scheme. You're seeing the system, no matter where it starts from, here or here, it will do what it needs to do. When it gets to the correct position in anatomical morphospace, that's when it stops. So you can think about this as a kind of anatomical homeostasis. Salamanders aren't the only ones to do this. Humans regenerate their livers. Human children regenerate their fingertips. And deer, large adult mammal, regenerates its antlers every year. So up to 1.5 centimeters of new bone per day, including vasculature, innervation, blood vessels, and so on, up to a very large structure. And they can do this. So this is, we can talk later about why it is that humans seem to have less capacity than some amphibians. But what I've just shown you is this idea that this multi-scale architecture that we are made of, so cells and tissues and organs, are able to navigate their anatomical state space, so get where they're going despite various injuries and perturbations and so on. And so what we're going to do now is talk about how that happens. How do the cells know what to make? Right? How do they know what a correct salamander limb is, for example, and how do they communicate with each other to make that happen? And for that, we're going to talk about something called bioelectricity. So let's just remind ourselves of what the problem is that we're facing here. Here is a normal tadpole. So here are the eyes, here are the nostrils, the mouth is here, the brain, gut. And so these tadpoles are supposed to become frogs. And in order for a tadpole to become a frog, it has to rearrange its face. So the eyes have to move, the jaws have to move, everything has to move around. And so it was thought that this was a hardwired, i.e. a very mechanical and simple process whereby, well, every tadpole looks the same, every frog looks the same. So all you have to do is somehow encode the right direction and amount of movement for all these organs, and then they'll end up in the right place. So we decided to test that. And this is very important for us. All of these estimates of intelligence, you have to do perturbative experiments. You can't just guess, and you can't have philosophical pre-commitments to it, and you can't just assume from observational data. You have to do experiments. And so what we did was, in line with James's idea that you look to see how the system reacts to barriers and perturbations, we made so-called Picasso tadpoles. So everything is in the wrong place. The eye, it's all scrambled. The whole face is scrambled. The eyes on the back of the head. The jaws are off to the side. It's a complete mess. And if this was a hardwired process, everything would end up in incorrect locations as the systems moved their normal amount in their normal direction. But that isn't what happens at all. These animals give rise to very normal looking frogs. And so what happens is that genetics doesn't specify a set of hardware rearrangements. What it actually gives you is hardware that executes an error minimization scheme. It executes a problem of reducing error from a particular set point. And so this is the story that you see in textbooks, which is very much focused on emergence and complexity. This idea that there are local rules, so genes interact in gene regulatory networks, they make some proteins, proteins interact via physics and so on. And eventually, out of lots of simple rules comes something very complex like this. Now, this is all true. Of course, this absolutely does occur. But it's only part of the story. That feed-forward emergence, right? The idea that complexity will emerge from the execution of local rules is only part of it, because when you deviate the system from that target morphology, and this could be with injury, it could be with teratogens, mutations, whatever, then some very interesting new activities will kick in, both at the level of physics and genetics, that are basically error reduction loops, and you end up with a process of a kind of anatomical homeostasis where they will try to reduce that error and get back to where they're supposed to be. Now, on the one hand, this is familiar. Biologists know all about feedback loops, of course, but there's a couple of interesting and novel things here. One is that typically homeostatic loops in biology are a scalar. In other words, a single number. It's a hunger or pH level or temperature or something like that. It's a single number. Here, that's not the case. You really have to know something about the complex shape of this organism to be able to correct after different kinds of damage. So that's interesting. So the set point of this homeostatic process is some kind of a complex data structure, but also we're not encouraged, especially in molecular biology and cell biology, we're not encouraged to think about systems trying to do things. We're not encouraged to think in a goal-directed way. We're told to think about chemistry and different ways that these subsystems interact with each other, and then what emerges, right? The science of complexity. We're not taught to think about models in which the system's actually trying to achieve some sort of goal in a problem space. So this weird way of thinking about it makes a very strong prediction. It says that if this is true, we ought to be able to find the biophysical encoding of that set point. How does it know what the right pattern is? Well, it has to be encoded somewhere. And if we were able to find it, we could do the same thing that you do with thermostats or with any other kind of cybernetic kind of system, which is if you understand where the set point is encoded, you can learn to decode it and you can rewrite it, and then let the system do what the pattern says. In other words, we wouldn't have to intervene back here, which is what most of the current molecular medicine approaches are. So you want changes up here at the system level, but all your tools are down here, right? Molecular pathways and things like that. And working this backwards is incredibly hard, if not impossible. And so that's a big advantage. If we could leave all this in place and simply reset the set point the way you do on your thermostat, you don't rewire the system. You just change what the system thinks of as the correct set point. That would be of high advantage. And so this is what we've been doing for some time now, is testing that hypothesis. Can we find, decode, and rewrite the set point of this homeostatic system? Now, in thinking about what might it even be, how would you encode a complex anatomical or geometric shape in living tissue? And one immediately thinks of the one example where that's not controversial. So in the brain, we know that in the brain, there's this hardware made up of cells in the network, which have these little ion channels in them. So they pass ions back and forth, and they get a voltage that they can then share with their neighbors through these kind of gap junctions or these electrical synapses. And that hardware drives a very interesting kind of software. And here you can actually watch. This group did this amazing video of electrical activity in the zebrafish brain. What happens in the brain, of course, is storage of complex patterns as goal states, and then control of the body to try to reach those goal states. So we know it's possible in biological tissue to store set points and to work hard to try to get to them. And there's this process of neural decoding. There's this program that research program that seeks to read information like this and extract from that all of the memories, the preferences, the goal states of the animal. Now, the remarkable thing is that trick, that incredible trick of having an electrical network store and work towards specific goal states is not unique to the brain. Evolution discovered it about the time of bacterial biofilms, and every cell in your body does this. Every cell has ion channels. Just about every cell has gap junctions that it can form with its neighbors. And so one can ask, what did your body think about long before it had a brain and muscle to move through three-dimensional space? What was this computational medium used for? And we can try to decode it, right? That's a very parallel kind of idea to what neuroscientists do, except outside of the nervous system. So we developed some of the first tools to read and write this information outside the nervous system. So the first is a set of voltage-sensitive fluorescent dye approaches that you basically soak your samples in, and they tell you where all the voltage gradients are. So this is a video of an early frog embryo made by my colleague, Danny Adams. And when she was a postdoc in my group, she worked out a protocol by which we could use these voltage dyes to see all the electrical conversations that these cells are having with each other. Then there's a lot of computer modeling that we do in simulation to try to understand how does the expression of ion channels in these cells give rise to specific patterns, and then what happens to these patterns when the tissue is altered and so on. I want to show you an example of two of these patterns. One is, we call this the electric face. So here's a time lapse. Again, this is a voltage imaging dye. It's a grayscale instead of a pseudo-colored. Well, what you see here is that at one frame of this movie, this is what the bioelectric pattern looks like. I'm showing it to you because this is one of the easiest, most obvious patterns to understand. It literally looks like a face. Here's where the eye is going to go. Here are the placodes. Here's where the mouth is going to be. We're literally looking at the pattern memory of these cells that are telling us what it's going to build shortly thereafter. And we know this pattern is absolutely required to have a normal face, because as I'm going to show you in a minute, when we perturb that pattern, the gene expression shifts and thus the anatomy shifts. Now that's a normal pattern. Now there's also a pathological pattern where if we inject a human oncogene into this tadpole, then these cells will eventually form a tumor. Eventually it metastasizes. But long before that, you can actually see, using this voltage dye, you can actually see these cells have disconnected from the electrical network here. They're going off on their own. And as far as they're concerned, they're amoebas in the big wide world. They're just treating the rest of this body as outside environment. And you can track this. So this is, we're of course working on this as a kind of cancer diagnostic tool, much like this is a birth defect monitoring tool. Now tracking all of these things is important, but even more important is the ability to rewrite them. So it's not just about reading these bioelectrical patterns, we need to alter them. And I want to be very clear that when we alter these things, we are not using electrodes. There are no magnets, there are no waves, no frequencies, no radiation, no electromagnetic fields. What we are doing is exploiting the natural interface that these cells are using to control each other, and that is the ion channels on their surface. So all of these cells expose this amazing interface to each other and to us, which are ion channels and gap junctions. And we now have all the tools of molecular physiology from neuroscience to control them. We can open them, we can close them. You can do this with drugs. You can use this with light, as in optogenetics. You can do it with genetic modification of the channels themselves. There are many ways to do it, but it's quite specific. The idea is, can we open and close these channels to induce a very specific pattern? And the answer is yes, you can. And I'm going to show you a few things that happen when you do that. First of all, here's an early frog embryo. And what we've done here is to inject some RNA encoding potassium channels. And what that RNA will do is set up a voltage, a local spot of voltage that looks just like that eye spot that I just showed you a minute ago on the electric face. And if we do that in some cells that would otherwise have been gut, then what these gut cells will do is change what they're working on and instead build an eye. So if you section through these eyes, they can have all the right lens, all the right components, lens, retina, optic nerve, all that stuff. And so from this, you see a couple of interesting features. One is that the bioelectric pattern is instructive. It's not about generalized damage. It's about telling cells what to do. If you move that eye spot to, for example, where the gut should go, then these are the cells that will make an eye. So the bioelectric memory pattern is in fact instructive for anatomy. That's the first thing. The second thing is the modularity. We didn't tell these cells how to make an eye. We didn't tell all the stem cells what to do, where to put the optic nerve, where to put the retina, and so on. All we did was give a very simple signal that basically just says, make an eye here. Everything downstream is the competency of the responding medium. It's the responding cells that know what to do when they get that signal. So it's a very high-level subroutine call. If you do any coding, it's basically a subroutine call. And the last thing that I find really interesting here is that if you look at your developmental biology textbook, what it will say is that only these cells up here in the anterior neurectoderm are competent to become eye. And the reason they say that's their estimate of the competency of the tissue. The reason people say that is because they've probed it with a master eye gene called PAX6. And if you misexpress PAX6, indeed, you can only get eyes up here and nowhere else. And so using that tool, it reveals to you a lack of competency in the rest of the body. But that's not the right tool. If you use bioelectric signals, in fact, you can get eyes anywhere. We've gotten them on the tail, in the gut, and anywhere you want. And so that teaches us an important thing, to be humble about the competencies of the material we're dealing with, because it's only as good as we are in giving it the right prompt. If you do this kind of thing, you can make ectopic inner ear, so otocysts here. You can make ectopic hearts. You can make ectopic forebrains, new legs. So here's our nice six-legged frog, and ectopic fins. Now that's kind of weird. Tadpoles aren't supposed to have fins. We'll talk about that in a minute. This is more of a fish thing. So we've been using this kind of idea to give high-level anatomical signals to redirect where cells are going in that space. And one obvious application is in regenerative medicine. So here are some frogs. When you amputate their legs at this stage, normally nothing happens. 45 days later, there's basically nothing. We have a cocktail of ion channel modifying drugs that immediately induces an MSX1 positive blastema. It induces, you got some, by 45 days, you've already got some toes, you've got a toenail, the most distal part, and eventually a perfectly respectable leg. And it's touch sensitive and it's motile, so it's functional. Now, again, the way this works is that we only intervene in the system for 24 hours. So for that first initial period, and then we don't touch it again. The record, our longest experiment is 18 months. So during those 18 months, a full-on adult frog regrows their leg when treated with a particular cocktail. And we don't touch it during that time at all. This is not about micromanagement. This is not about telling stem cells what to do or scaffolds or any of that. It's about convincing the cells on day one that they're going towards the leg building region of morphospace, not the scarring region of morphospace. And here I have to do a disclosure, which is that Dave Kaplan and I have this company called Morphoceuticals, where we are trying to move that approach now from amphibians to mammals, and we are doing work to try to get in eventually, hopefully, to get in the clinic with a mammalian limb regeneration application delivered by this wearable bioreactor, which is what David's lab does. Okay, so I want to shift gears to really nail this idea of these bioelectric patterns as kinds of memory. And to do that, I want to talk about a different organism. This is called planaria. And so these guys are these very cute little worms. They have a true brain. They have a central nervous system. They have lots of different organs and tissues, same neurotransmitters as you and I. And they have this amazing property that if you cut them into pieces, and the record I think is like 276 pieces or something like that, you cut them into pieces, every piece regrows into a perfect little worm. So that's amazing. So every piece gives rise to a complete correct anatomy. And by the way, not only are they cancer resistant, but they are actually immortal. So there's no such thing as an old asexual planarian. And they also have an extremely messy genome, which is also another interesting thing that we can talk about. But they have this amazing anatomical fidelity. And so what happens is that if you amputate, let's say, the head and the tail, and you leave this middle fragment here, you might ask, how does this fragment know how many heads it's supposed to have? And that's the question that we asked. How does it actually know how many heads it's supposed to have? And it turns out that there's an electrical circuit that we characterized. And this electrical circuit is what tells it how many heads it's supposed to have. And so this is what the voltage looks like in that fragment. You can see that it says 1 head. Now, what we're able to do is take that animal and rewrite this electrical circuit to say 2 heads. This is kind of messy still. The technology is very much still being worked out. But we can say, no, you should have 2 heads. And what will happen is that if you then cut this animal, it will, in fact, have 2 heads. So this isn't Photoshop. These are real animals. Now, the amazing thing is that this voltage map is not a map of this animal. That's really critical. This is not a map of this 2-headed guy. This is a voltage map of this perfectly anatomically and molecularly normal animal. Only if you cut him does he then make 2 heads. In the absence of that injury, he just stays how it is. And so a single planarian body, a normal 1-headed planarian body, can store 2 different representations of what to do if you get injured in the future, 2 different ideas of what a correct planarian looks like. So there's a couple of interesting things here. First of all, this is basically a counterfactual, right? This idea of brains being able to do this amazing time travel where you're able to think about things that are not true right now. So you're able to have memories of the past, anticipation of the future, right? So that's mental time travel. So a very basic primitive version of that counterfactual is this ability to store a pattern of what you will do if you happen to get injured in the future. And so this is why I'm saying that this is that ability to rewrite that set point. We literally have rewritten the representation of how the pieces know what the correct anatomy is. And once you've rewritten it, well, that's the only thing they have to go on. There's nothing else for them to compare it to. And so then this is their new normal. Now, I call this a memory because it has all the properties of being memory. In that, if you take one of these 2-headed worms and you cut off the primary head and you cut off this ectopic secondary head, and you just leave, in plain water, you just let them regenerate, what they will do is they will continue being 2-headed, even though they have a normal genome. We haven't touched the genome. There's no genomic editing here, no CRISPR, no transgenes, nothing like that. The memory is in the bioelectrical circuit. They literally remember how many heads, and like any good memory, if you rewrite it, it keeps. And so you can keep cutting them as far as we can tell forever, and they will stay 2-headed. Here are these animals. You can see them hanging out. And so this has all the properties of memory. And again, think about the role of the genome here. If I were to ask you where is the number of heads in a planarian encoded, and you said it was in the genome, you'd be wrong. What the genome does encode is hardware that by default starts out encoding the state that's as one head. But it's not determined, it's not nailed down by the hardware, because you can change it. You can change it in situ without actually changing any of the genetics. And that's just the tip of the iceberg of information here that's generated physiologically in that hardware. Now we are not the only ones that can hijack this electrical system. Bacteria can do it too, and this is a study that we did in collaboration with Ben Wolf and Catherine Williams. Kate's was an undergrad in our lab, and what she found was that there are bacteria that live natively. They're part of the microbiome. They live natively on these planaria, and when we interfere with the relative proportions of the different types of bacteria, they can exert a strong influence on their host, for example, to make their host two-headed, or to make radical changes in their visual system. So this is the normal here, the eye spots here, the optic nerves, radical changes in that visual system, all driven by, again, no genetic change, but all driven by signaling from the bacteria that are able to interface to the native controller that sets things like head number, head shape, and so on. So what we're doing now is we're trying to unify two very different approaches that people take to problems like this. On the one hand, the state space of the biophysical circuit that shows you all the different ways that it could be, and some ideas from connectionist machine learning and dynamical systems theory that try to understand how do networks store information, how do they recover from partial input, and so on. And so these we're trying to unify, and some of that is described here. Now, interestingly, it's not just about head number. It's more general than that. It's about head shape as well. So if we take this nice triangular headed planarian, amputate the head, and then confuse the bioelectrical circuit about what it should be doing, then you sometimes get flat heads like this P. felina, or you get round heads like this S. mediterranea. These creatures are between 100 and 150 million years distance from this guy. Again, remember, no genetic change here. Not only the head shape, but actually the shape of the brain and the distribution of the stem cells changes to be just like these guys. So this hardware, this perfectly normal genetic hardware, is happy to visit other attractors in that morphospace that belong normally to some other species, but that hardware can go there, no problem. So you're starting to see this idea of reprogrammability. This is what I said at the beginning. The biological tissue has the ability for you to call large scale subroutines with very simple triggers, very simple signals, and then everything else gets handled underneath. It has the ability to remember and to store new patterns that you write into it using this physiological interface, and it has the ability to visit different attractors other than what the default of the hardware normally is. Now, you can even visit some attractors that you wouldn't say belong to planaria at all. So you can go further, and we've made these spiky forms like this, these cylindrical things that are not even flat, and then of course the hybrid these kind of hybrid shapes. And so you can think about exploring, and this is something that Darcy Thompson wrote about as early as 1917, explore the latent space around any particular form to see what else it's actually capable of. And I think this space is, if we were doing a bioengineering talk, I would show you that the space is actually quite enormous and fascinating. The last story I want to tell you on the bioelectric front has to do with repair of birth defects. So and a new way to address the repair of very complex structures. So here's a frog brain. You've got the forebrain, midbrain, and hindbrain. You can see it comes in two halves, left and right. And if you expose these embryos to various kinds of teratogens, and that might be nicotine, alcohol, various mutations, you can get characteristic defects like this, for example. And so we sought to ask a question. How could we repair something complex like this? And to do that, we created a model, a computational model of the bioelectric pattern that normally tells this brain what shape and what size it should be. And the thing about this bioelectric pattern actually is that it has this very characteristic bell curve shape, and flattening it out either up here or down here both has the same effect. It gives you terrible brain defects. It's very important that this particular shape in the electrical pattern be there. And so we simply asked the model then, okay, but if the pattern is wrong, what channels could we open and close to get back to that pattern, right? So it's the idea is using a predictive computational model of the bioelectrics to say, how do we fix that memory, the pattern memory of what you're supposed to be? And amazingly, the models made a suggestion about a very interesting channel called HCN2. And when we tried it to test the predictions of the models, so we basically upregulated HCN2 in these creatures. Here is a normal brain. Here is and I'm showing you actually what I consider to be the most impressive or the worst kind of teratogen. This is a genetic mutation. So this is a mutant in a gene called Notch. Notch is a very important neurogenesis gene. So with that overactive Notch mutant, there is no forebrain. The hindbrain and midbrain are a bubble. These animals have no behavior. They just lay there. And so this is a very severe defect. And so we asked, could we possibly fix this? Could we repair this state? And it turns out that if you upregulate HCN2 channel activity, just like the model said, either by injecting new HCN2 RNA or by using drug openers of existing HCN2s, and these are human-approved anti-epileptics, that if you do that, you get back to the correct brain. In fact, these animals have not only the right brain shape, but they have the right gene expression in the brain, and they have their IQs back, because we can do behavioral testing, and they learn in a way that's indistinguishable from controls. Now, I find this remarkable because it turns out that using the bioelectric interface guided by a computational model, you can sometimes fix certain hardware defects in software. I'm not claiming that will always be possible, but in this case, a hardware defect, a broken Notch signaling pathway, can be overridden by giving these cells a reinforced memory of what they should be doing, right? So this is one of many examples where the physiological state trumps the genetic state. So more generally, what we're doing now is setting up a system like this, where you could go from the initial issue of, okay, I have an incorrect pattern, that might be a birth defect, it might be a tumor, whatever, and you know what the correct pattern is because we're working on the physiomic data sets, and you know which channels are in your tissue of interest, because that already exists, because all this stuff has been profiled and online. And so if you integrate the simulator, you can ask which of those channels can be used to get the pattern back to normal. And so this is just the beginnings. You can actually play with this. It's online. This is the beginning of the interface, where you could pick different tissues, either normal or transformed, and then see all the channels that are your potential targets, so-called electroceuticals, because there's a huge database of drugs that target all of these channels. Many of them are human approved. So this is what we're looking at in the future, is the use of computational platform guided electroceuticals for birth defects, for regenerative medicine, and for cancer. I will mention, just for this audience, I will mention that the bioelectric system also affects immune function. So we in particular studied the innate immune system, and we found that survival of embryos when they were challenged with a pathogenic bacteria is much improved if you hyperpolarize the tissues with specific drugs or in fact RNAs. And some of that functionality is mediated by serotonin. So if you put a serotonin transporter dominant negative in, you can abrogate the activity. And when we looked at using next-gen sequencing, so RNA-seq, we found that changes in bioelectrics alter many different targets that belong to the inflammation, infection, and other kinds of immune-relevant endpoints. And in fact, we also found, this is kind of interesting, we found that the presence of the brain is an important component of that loop. So in the absence of the brain, and the nice thing about these model systems is you can remove the brain, the body continues to develop. It's okay, they're completely viable. But what they do have is decreased ability to fight off infections. So there's this really interesting interplay, and I don't have time to go into all the details, but you can catch that here, and even some information about macrophages and the adaptive immune system. But there's this really important interplay between the brain and the non-neural bioelectricity of other cells, and the immune response that leads to either tolerance or susceptibility, or to clearance of bacterial infection. And one of the things that we found, this is a cross-section through a frog embryo, is that if you inject fluorescent bacteria, so this is pathogenic E. coli made fluorescent by expressing GFP, this pattern is very familiar to developmental biologists. This is exactly where neural crest cells travel in the body to make various organs. So the bacteria are following these same paths that native cells follow during morphogenesis. There's much more to learn about this. We're just beginning to scratch the surface. So for the last few minutes, I want to kind of bring it back and think about what this means for questions of biomedicine, but specifically about infection and things like that. And I want to focus on this notion of hacking as a fundamental concept. What we've seen up until now is that cellular collective, so groups of cells, use bioelectricity to keep collective memories of what they should be doing, specifically in anatomical space. And they have the ability to respond to various cues, for example, novel bioelectric signals, and take journeys in that morphospace and build various organs and repair organs and things like that. And so the idea is that we really have a collective intelligence here. It's held together by bioelectricity, very much like your collective intelligence of your individual neurons is held together by the bioelectricity of your brain and nervous system. So let's think about how that looks in other spaces. So these are some examples, and I'm sure you all know more about this than I do. But there are many examples in which parasites actually hack behavior. So this is toxoplasma that is known to affect the behavior of the animal hosts that it's in and the humans. So it does things like lower risk aversion and things like that. And then of course there's this zombie ant fungus, which again controls the body of the ant and forces it to take various behaviors that facilitate the fungus life cycle and so on. So the idea is that creatures hack each other's policies for navigating three-dimensional space. There are lots of parasites that do host-altered behavior. But that's not all, because these parasites can also hack the behavior of life in anatomical and physiological spaces. And what do I mean by that? Well, here's development, and we really the development's amazing, but it really lulls us into a false sense of understanding, because we look at this, the reliability of this process. Here's an acorn, here's an oak leaf. And we tend to think that we know what the oak genome does. It takes you on a particular path in morphospace from here to here. This is what happens 100% of the time. This is what happens. So that's what we think that the hardware encoded by the genetics, we think that that's what it knows how to do. But one thing we would never have guessed is that these exact same cells are also capable of forming this or who knows what else, because there's a little hacker. In this case, it's a wasp that lays down an embryo, and that embryo signals to the plant cells. It doesn't build this thing. These are not wasp cells. These are plant cells. It puts out some signals that convinces the cells of the leaf to build this kind of crazy structure. And from looking at that leaf or from looking at the oak genome, we would have absolutely no idea that these cells are even capable of doing something like this, taking this journey in morphospace, until we found that it was prompted by this. So this idea of we call it bioprompting, this idea that if you're dealing with a reprogrammable competent material, it's not all, you don't have to micromanage it. You can find some very high level signals that are going to exploit the competencies of that material to build specific things and to take other journeys. And so the key questions here are something like this, that as a multi-scale biological system, how do you know when you are being hacked? How do you know where the border is between you and the outside world? Which signals are yours? Which signals are coming at you from outside? And when you're born, what are you? What sensors do you have? What effectors do you have? You really don't know. And there are many examples in psychology as well as physiology of disorders in the ability to set a border between you and the outside world. Right? There are lots of patients who have trouble figuring out whether various impulses they have are coming from them or from the outside. And there's this idea. Think about the difference between learning versus being trained. Yeah, when you're learning, the implication is that you're the boss and you're functioning in this sort of dumb environment, and it's up to you what you're going to learn from that environment. But when you're being trained, the situation is similar on one hand, but on the other hand, there's another agenda on the other side of that environment is no longer zero agency, right? There's another agent there that's serving their agenda by training you in a particular way. And so being able to tell which signals are yours, which signals are coming from your own body parts, and which signals are coming from a parasite means that it's really important to know the boundary computationally, not just physically, the skin or whatever, but the computational boundary between you and the outside world. And for biomedicine, the other thing that's really important is really to get the buy-in from cells on whatever signal you're trying to send, because oftentimes if you trigger this kind of resistance, then what you're going to get are things like drug habituation and failure of the ability to reset to affect any kind of permanent cures. You can manage symptoms while you're giving a drug, but as soon as you take the drug away, it goes right back, or in fact, sometimes it gets worse. Most of our drugs have that property. So the idea to avoid resistance is to change the set points, not to micromanage the symptoms or the current state, but to actually change what it is that the collective of the cells is trying to do. And so let's just think about this. How do you set the boundaries between self and world? This is a schematic of an embryonic blastoderm. Let's say 50,000 cells. We were all something like this once, flat sheet. When you look at this and you say, oh, there's an embryo, what are you really counting when you say there's one embryo? What is there one of? I mean, there's 50,000 cells. What is there one of? Well, what there's one of is alignment, both physical, be in the sense of planar cell polarity, but also in anatomical alignment of all of these cells towards a specific goal. All of these cells are working together to build one specific thing, meaning they all are going to take the same journey in that anatomical space from a single cell to a particular complex anatomy. And this is actually quite similar to a problem that you might have when looking at a brain. If you didn't already know what a human was, and somebody showed you a 3.5 pound or whatever it is of brain tissue and said, how many individuals do you think fit in there? How many cognitive selves do you think could fit in there? We have absolutely no idea. We don't know what the density of a typical of a neural medium is for a typical human type of cognition. We really don't know how many you can fit in there. There's all kinds of interesting clinical work on that. So the same thing is true of the embryonic blastoderm. When I was a grad student, I used to do this experiment. Take a little needle and scratch. This is a duck embryo. Scratch put a little scratch in that blastoderm, and for about 4 hours until they heal back up, each of these little islands doesn't feel the rest, and it decides that it's the embryo, and you get conjoined twins like this, or triplets. This means this is really interesting. It means that when you look at one embryo, there's the question of how many cells or how many individuals are in there is not fixed by the genetics. It might be 0, it might be 1, it's usually 1, but it might be actually maybe up to half a dozen, depending on what these cells decide to do and how the collectivity works. And because every cell is every other cell's neighbor, right? Every cell is somebody else's neighbor. Then in this kind of joined blastoderm, you see that each individual has to sort of decide, where do I end and where does the other guy begin? That idea is right there from embryogenesis. That setting up that border between you and the outside world has to be calculated in the beginning. And some cells are actually confused, they're not quite sure, and that gives rise to actually laterality defects in conjoined twins. This is something that we worked out back in '96 or so, why it is that conjoined twins have laterality defects. It's because some of these cells don't know, am I the right side of this one or am I the left side of that one? So what happens is that, and this is just the last couple of ideas here, what happens is that when cells get together, they are able to inflate or enlarge their cognitive light cone in space and time. They're able to perceive more signals. They're able to have larger computational capacities. And this is what evolution has given us based on the ability for using this bioelectrical and other interfaces to scale up our goals from the tiny little goals of these cells, so metabolic and so on. They scale up to these very grandiose goals of keep maintaining a limb and repairing it if anything happens to it. But that has a failure mode, and that failure mode is cancer. And when these cells disconnect from the electrical network, they can no longer support these complex representations of things they should be doing. They go back to a single cell amoeba lifestyle, basically proliferate and migrate to wherever life is good. And this is glioblastoma. And so on this kind of weird idea that cancer cells are not more selfish than other cell types, right? Lots of game theory model cells as being less cooperative or more selfish. They're not more selfish. I think it's just their selves are smaller, whereas the self here is quite large and it maintains this whole thing. These have a little tiny amoeba-sized self. And so using that crazy idea, we've actually developed a therapeutic around inflating the boundary between self and world for these transformed cells, not killing them, but actually co-injecting ion channels that force them to remain in electrical connection to the rest of the tissue. And here, these are nasty oncogenes like KRAS mutations and so on. But if you manage the bioelectrics appropriately, even though the oncoprotein is blazingly expressed, like look at it, in fact, it's all over the place. There's no tumor. This is the same animal. There's no tumor, and that's because it's not the genetics and it's not the genetic it's not the oncogene that drives the phenotype. It's the physiology. It's the decision of the cells in a collective that they're not going to be acting as amoebas. They're going to be making nice skin, nice muscle, and so on. And so remarkably, that process actually involves bacteria. It involves butyrate made by bacteria. So here's yet another, and we study that in this paper. It works at a distance from where the oncogenes actually are because there's this signaling used by depending on a butyrate transporter that's part of this whole process. So once again, the microbiome is key here. And so I'll just end here by pointing out that this idea of cells hacking each other is ubiquitous. We are only able to do these things because that is what the cells expect anyway. They have an interface that allows them to hack each other. And that when, come back to this example, when we inject an ion channel to force these cells to build an eye, if we only hit a few of them, so this is a cross-section of a lens sitting in the tail of a tadpole, and the blue ones are the ones we injected, what do? Well, they hack all their neighbors to cause them to participate with them because there's not enough of them. They know there's not enough of them to make a proper lens, so they recruit their neighbors. And that, of course, is seen in other kinds of collective intelligences like ants and termites, where a few of them will take on a project. There's not enough of them, so they will recruit their buddies to help them. So the cells are constantly hacking each other and trying to convince each other to do new things. Sometimes they cooperate, sometimes they compete. This is all discussed here. Actually, competition between your body parts is quite important. And so all of this focuses on not a bottom-up approach where we talk about micromanaging specific pathways, but a top-down approach where we ask, what does the collective remember? What are the goals that it's trying to reach? Where are the set points encoded? And how could we hack them by rewriting these set points and talking to them at a very high level? So this is kind of my layout of biomedicine, where most of what's going on is here, these kind of bottom-up interventions that are trying to micromanage certain things. And all of this is what I think of as future medicine, this idea that it's going to look a lot more like a kind of somatic psychiatry and a lot less like chemistry, because all the key models in this field are going to be around information. They're going to be around what do the collective agents at different scales, what do they believe? What are their what's their calculus of decision making, and so on. And you can read more about that here. So lots more papers if anybody wants to dig down in some of the philosophical aspects about what machines are and what cognition is, and so on. And I'll just stop here, and I want to thank the people who did all the work. So J.F. Paré, who did the native immune system bioelectrics, and Celia Herrera Rincon, who did the brain and immune system work. Vaibhav Pai did all the ectopic eye work, and the planaria work was done by Johanna Bischoff and Fallon Durant. Danny Adams did the voltage dyes, and Juanita's work and Brooke are working on the bioelectrics of cancer. So I'll thank our various technical support that we have in the lab, our many amazing collaborators, our funders here that have supported the work that I told you about. Two disclosures, so Morphoceuticals and Astonishing Labs, which are two companies that support some of our work, and most of all, the model systems, because the animals really do all the hard work. So thank you very much, and that's it. I'll take questions.